Hello, everyone. Welcome to MedCity Invest Conference and our last Pitch Perfect track of the week. Today, we're welcoming 5 diagnostic startups to compete. But before we get to the actual contest, I wanted to take a few minutes to sort of introduce MedCity News to you. I know some of you have already seen the slides and have probably got them memorized by heart, but indulge me this one last time to talk a little bit about the company and our editorial coverage. So MedCity was founded in late 2008 to cover innovation in healthcare. We are a national publication drawing nearly 2 million page views monthly, and we are seeing growing engagement now that everyone is interested in healthcare. We cover biopharma, digital health, medical devices, diagnostics, as well as hospitals and payers in the context of the industry's overall transformation. We also host conferences focusing on investment, population health, precision medicine, digital health, and patient engagement. We accept outside content through our influencers program. We also launched a new podcast during the pandemic called Pivot. I think this word needs no further explanation given that we're all doing it in our own personal lives, whether it be for kids that we have to shepherd through distance learning or in our business lives, such as MedCity, having to go from in-person conferences at the Ritz-Carlton in Chicago to this sort of virtual online Zoom forum. So if you have any suggestions for interesting speakers, please send them my way. You have my email address up here, and please listen to all the episodes. We've had some really interesting guests, so I urge you to go to our website and listen to some of the podcasts. I wanted to take a moment to thank all our sponsors, JP Morgan, Unity Point Health, which actually sponsored one of our pitch tracks, BioEnterprise and TriNet Health. Obviously, without them, we wouldn't be able to host this conference. A big thank you, as always, to our partner, MHIN. It is a nonprofit organization comprised of more than 50 active healthcare investment firms with offices throughout Middle America. We also have a new actually not new, but exclusive presentation that we did by talking with Pamela Allsworth, Managing Director and Head of VC Coverage at JP Morgan. She had a very interesting take on how the pandemic is changing deals and deal making, and also some much needed advice for startups on how to survive this pandemic. So I wanted to go a little bit over the process of how we're going to conduct this contest today. Obviously, we have 5 very interesting diagnostic startups. As each startup comes to present, the executive summary for that company will be presented in the group chat, so feel free to access those. Each startup will have 10 minutes, 4 minutes to pitch, followed by a 6-minute Q&A with judges. Time will be enforced strictly. I'll chime in with 1 minute left when you have another minute left, and then I'll say time's up when the time is up. At the end of the 5 presentations, we will do a quick online poll to determine an audience favorite from today's track. And then the winners, based on the judges' opinions and scores, will be announced Monday on medcitynews.com. Now let's meet our judges, Navneet Alag, Derek Mazur, and Chang. Navneet is VP at HIG BioHealth Partners and focuses on investment opportunities in the healthcare sector, including biopharma, medical devices, and diagnostics. Previously, Navneet was an engagement manager at McKinsey & Company, where he led strategy development for pharmaceutical and medical device companies on R&D, mergers and acquisitions, and commercial launch strategy. Prior to McKinsey, Navneet worked as a research scientist at Genomtec. Welcome, Navneet. Next, we have Chang. Sean is currently an investment manager at Phillips Ventures, a portfolio of promising health tech companies. He's interested in early stage investment opportunities in digital health, medical devices, and therapeutics. Previously, Sean held positions at the Boston Consulting Group, the US FDA, and NASA. Sean also serves on the board of directors of the Professional Center for Child Development, board of advisors at the Johns Hopkins University, and the advisory council of the World Economic Forum's Global Shapers community. Welcome, Sean. And last but not least, we have Derek Mazur. He's senior associate at Santee Ventures. He joined the company in 2014. Prior to joining the firm, Derek was with Houlihan Lokey in their healthcare mergers and acquisitions practice based in Chicago. Before Houlihan, Derek was in investment banking division at SunTrust Robinson Humphrey based in Atlanta, where he focused on a variety of syndicated and leveraged finance, capital markets, and financial advisory engagements. Derek currently serves as a board observer at Iterion Therapeutics, a clinical stage oncology company, and Remedy, an on-demand urgent care service that brings the doctor to you. Welcome, Derek. And now let's meet our pitch perfect finalists. We have Exogene Bio, Genome Tech, Atom Bioworks, SEngine Precision Medicine, and Amplified Sciences. Welcome to all of you. So we'll start with SEngine Precision Medicine. As its name suggests, it's a precision oncology company revolutionizing cancer diagnostics and therapies by pre-testing drugs on patient-derived organoids grown ex vivo utilizing patient-specific tumor cells. As a spin-out from the world-renowned Fred Hutchinson Cancer Research Center, SEngine is leveraging over 2 decades of R&D in diagnostics and drug discovery. The company is commercializing the PARIS test, a next-gen diagnostic test that predicts drug responses, integrating knowledge of cancer genomics with organoids, robotics, and AI-driven computational tools. Carla Grandori, co-founder and CEO, will present first. Take it away, Carla.
Thank you. Okay. I hope you can see the first slide.
Yes, and your time starts now.
Yes. Excited to be here to tell you about our company latest development. Every year, 9 million people die from cancer, and a quarter alone in the United States. Despite the progress made by the adoption of DNA sequencing, this information still fails to provide personalized treatments for the majority of patients. We have learned that every individual cancer is a unique mosaic of molecular changes. I actually have those mosaics in the back in my office. Thus, it's very difficult to translate this complexity in therapies for individual patients. We have developed a search engine for personalized cancer treatments. It's a next generation diagnostic because it's functional versus descriptive that is DNA sequencing. It combines the latest advances in maintaining cancer alive ex vivo as 3D organoids, the accuracy of robotics to test multiple drugs simultaneously, and unique analytics to enable identify targeted therapies for individual patients. We named the test PARIS in analogies to the mythological hero that defeated Achilles. So the PARIS test delivers an actionable report to oncologists where drugs are ranked from the most exceptional responses down to drugs that exhibit resistance. It integrates and interprets the DNA sequencing information to prioritize actionable mutations. A recent analysis that we presented at the ASCO and ACR indicates that our test is at least 4 times more actionable than DNA-based assays and has a strong predictive power. We're collaborating with leading cancer centers to advance the PARIS test for clinical use using clinical studies and real-world evidence. SEngine has also created a powerful platform to advance and discover new drugs. As Dr. Siddhartha Mukherjee comments as he joins our scientific advisory board. Indeed, we have identified new drug targets for drug development and advanced this particular target, the WEE1 gene, that had already a drug. A co-founder of SEngine initiated a clinical trial that was successful with this drug for heavily metastatic head and neck cancer with incredible durable response in a large proportion of patients. In summary, our investment highlights and business model. The foundational discovery. We're from the Fred Hutchinson Cancer Center over 2 decades. We are planning to commercialize the Paris test, and it's already CLIA certified, and it's applicable to all solid tumors. We are also utilizing the platform, as Siddhartha commented, to accelerate drug discovery. We are currently raising a Series B investment to allow us to scale up the Paris test and execute the clinical studies. We'd like also to expand our headquarters lab and potentially create satellite labs across the country. We plan to also use the investment to accelerate drug discovery through pharma partnerships. I encourage you to look at our website to learn more about our experienced management team. And I'd like to conclude with a quote from Lee Hartwell, Nobel laureate, and a member of our board of directors. If I had cancer, this is the way I'd want to be treated. And now I'll take questions.
Great, Carla, thank you very much for that. I apologize, I mispronounced the name of the company, it's SEngine, but I also wanted to congratulate you. I don't think I have come across a biotech company or any company for that matter that is using Greek mythology as a name of their test. So that was kind of interesting, although Paris is a bit of a sneaky creature. Sneaky character. We'll go straight to questions. Let's start with Navneet, and then we'll go to Derek and Sean.
Great. Hi, Carla. Thank you for walking me through the story. This was very helpful and very well laid out. So appreciate that. Quick question, and I think you might be getting this pretty often. If I am a payer or even a physician trying to rely on these 3D organoid tests, figure out which treatment to prescribe. What evidence do you guys have other than retrospective? Is it all retrospective, or do you guys have even some smaller, more targeted prospective analysis where you predicted which therapies could respond and then you've seen those results come through? Or is that just what is in plan and currently it's all retrospective?
We already have prospective evidence. We haven't published that yet. It's about 22 cases, and we have 80% prospective evidence of positive predictions, and some are really astonishing. Actually, some cases are going to be presented at the Cholangio Carcinoma Foundation meeting, and we're also mentioned at the ACR. Patients that have exhausted standard of care, for which we find drugs. I'll give you an example, dasatinib, that induced a 9 months progression-free survival for a patient that was probably going to hospice care.
Fantastic, thank you.
Yeah.
Great, let's go to Derek.
Thanks, Carla. That was 4 minutes of pretty well and thorough pitch, so it's a nice job there. My question is more about the business. It seems like there's really two things going on with this. It's a diagnostic test that can be used for more effective oncology treatment, but it can also be used as a drug engine. So what does the monetization of those two business lines look like? And I also want to take a step back and ask, are those the two business lines that the company is pursuing right now?
Yes, they are. And as you can imagine, to obtain revenues from the Paris test is going to take a couple of 2 or 3 years at least until we have a large data set of clinical utility to obtain insurance reimbursement. So our major monetization now of the platform is through advancing drugs with pharmaceutical companies, both external candidate, but also candidate we have internally, always through partnerships. So.
Okay, thanks.
Great, we'll go to Sean.
Yeah, thanks very much, and much appreciated on the Greek mythology names. I do wonder who's Achilles in this scenario. So my question would be maybe along the same lines is on the CLIA certified LDT side, are you going after specific reimbursements already with established PLAs, or is it more create your own path which probably delays the monetization on that end a bit more. What's the roadmap there?
Yes, so that's where the Series B is going to be employed for, to have a very expert-guided path toward insurance reimbursement, and if necessary, going through the FDA route. We have one cancer type that has incredible unmet need. I mentioned cholangiocarcinoma, so we envision landing first on this more restricted use and then branching on to other cancer types.
We have a little more time left, so if anyone wants to ask a follow-up, please go ahead.
Sure. Since my venture capital hat is on, I think this is a pertinent question to ask. This kind of relates to my first question. Who do you think buys the company? One interesting observation is that you can have multiple partners with pharma. And then there's the diagnostic testing, and that will have some value of its own. But who do you envision the acquirer set is for something like this?
Can you repeat the last question? Who do I envision as
potential acquirers for this company and this technology?
Yes, so we are already attracting interest from obvious companies that are involved in precision medicine from the DNA sequencing angle. So that will be an obvious merge. But we are right now, because of also our portfolio of novel drug targets, we're looking to be a bit more advanced before we will look for the type of acquisition that will fit right. Yeah.
Okay, great. Thank you so much, Carla. I really appreciate it.
You're welcome.
We'll continue on with our next startup. Exogene Bio's goal is to enter the liquid biopsy market in order to simultaneously isolate exosomes and cell-free biomarkers using proprietary nanofluidic technology. This technology will then be integrated into an automated medical diagnostic device for the simultaneous detection of multiple classes of cancers using early-stage biomarkers contained within exosomes and in cell-free fractions of body fluids. The early detection of these cancer-specific biomarkers will offer patients more effective treatment options with fewer side effects while significantly reducing healthcare costs. Anand Kamath, CEO and founder, will present next. He will then be joined by Stephen Mikalicek, financial consultant, during the Q&A. Please take it away, Anand.
Can everyone see my screen?
Yes, your time starts
perfect.
now.
Thank you for having us this afternoon, and thank you, judges, for being here. The title of my talk is a novel liquid biopsy approach for the early detection of disease biomarkers. And before I get into the content of my conversation this afternoon, I just want to preface what these biomarkers are. And I'm going to be focusing on exosomes, cell-free DNA, cell-free RNA, and cell-free products. As you probably know, the liquid biopsy space has been fairly crowded. And the traditional techniques for liquid biopsy capture have been convoluted, slow, and laborious, and clinically irrelevant. And by that, I mean that if you're in a lab and you're using things like ultracentrifugation, immunocapture, filtration, or precipitation, it's just too slow to yield exosomes and cell-free DNA, RNA, and other biomarkers that can be subjected to downstream interrogations such as sequencing or other next-gen techniques. One of the things that we have also pivoted on since the COVID-19 pandemic is that the agnostic sensor that we've developed, which I'll discuss next, also allows viral isolation just as easily as exosomal isolation down to 20 nanometers in size. So in conjunction with our semiconductor partner on the East Coast, we've developed an agnostic chip that can separate exosomes, viral particles, and DNA, RNA from small volumes of either plasma, urine, or cerebrospinal fluid. Now, this technology is based on a geometric and architectural design, so it's agnostic, it's label-free, and it's continuous flow. It's also scalable, simple, and ready to deploy. Now, what we've developed is an asset. This is not a developmental project that we're looking for funding. The chip has already been developed and is protected intellectually by 19 US patents, 32 US patents and applications, and 16 international patents. The chip has been validated. We've done studies both in-house and in other centers. We've also had numerous publications have been published, and the footprint of this chip is that it's easily scalable and easily designable in terms of microfluidics or nanofluidics foundry. Now, if we look at just one portion of the application of this chip and the original intent of my talk, which was cancer, we're looking at a $56 billion annual market for diagnostics. Now, this includes patients who are newly diagnosed with cancer, people who are undergoing treatment, and people who are also undergoing preventative treatment. Now, these preventive tests like Cologuard and other occlusive tests are entering the market that are replacing traditional tests like colonoscopies, et cetera. And we feel that the liquid biopsy space, especially for diagnostic testing in cancer, is only going to grow. Now, in terms of deployables, we have a product that is ready, that has unmet need, and that has gained interest from large players in this field, including places like Thermo Fisher. We've raised $2 million out of a $4 million Series A round, and we're looking to complete the Series A round with the second part, which is the $2 million raise. We see this as a massive market, which is doubling at $27 billion per year. And the money we're seeking is to complete the development and commercialization of this nanofluidic technology. Our scientific advisory board encompasses people both in academia and industry, and includes people who are at large firms such as Rockwell Collins, GE Healthcare, Thermo Fisher, and academia, including places such as MIT and other institutions that have allowed us development of this technology. Our external collaborator is a Fortune 500 company, which has one of the most expertise both in semiconductor technology and artificial intelligence. I want to thank you for your time and your attention to this talk this afternoon.
Great, just on time. Thank you very much, Anand. We'll take our first question from Derek and then go to Sean and Navneet.
Great, thanks for that. So I understand it's a 4-minute pitch, but this feels like a lot of other technologies that I've seen. So I guess could you just expand upon why this is better than the standard of care or the competition?
Sure. So the reason why it's better, for one reason, is that it's label free. So it doesn't require any complexity. You can do this with compressed air in a lab with very limited resources. So if you compare things like ultracentrifugation, which requires large centrifuges, or you look at things like cartridge-based technologies, you're looking at more consumables, and you're looking at exosomes and DNA, RNA that have potential share-based problems. You're looking at actionable data that's better with this chip. And the ease of this is that you can use any particle, both in terms of a polymer like DNA, RNA, or a spheroid, such as a coronavirus particle or exosome, and do it all on the same chip. The second thing that is better about this is it's much faster. It takes 10 minutes of time, and it's much easier in terms of the individual who's essentially performing this assay.
Got it. Thanks for that.
Mm-hmm.
Let's go to Sean.
Thanks, Anand. Question from my end would be on the logistics side. So you refer to Cologuard, which has a supply chain that runs all the way to the, I believe, to the home.
Right.
Do you want to play in the technology space and then partner up, or extend along the value chain there, and then run the logistics side as well?
So I'm going to have Steve and Mikhailchuk jump in here just to kind of give his thoughts.
Yeah, I think the go-to-market space is in the clinical space because it's sort of easy to use and it's rapid. So you'll see this in blood centers and then information being sent back to the doctor for assessment.
Got it. Makes sense. And are you already paired up? I think there might be something in the excerpts that you sent out with some certified labs to do this. No, we're at proof of concept stage right now.
We have a minimum viable product, and we're seeking partners right now. We've worked with academic centers and industrial partners. I think the way we're trying to do it, Sean, is the first will be a phased rollout that addresses the research market, and then it'll go towards the clinical market. So lots of people have expressed interest in using the chip for their own downstream applications once they isolate either exosomes or cell-free biomarkers, and they're not interested in necessarily getting the value-added information. They want to pair it with their own next-gen sequencing, for example. And so they want to get the chip and they want to get the portable unit, both of which are ready for rollout. Ultimately, what we foresee is to create a medical device where this razor blade will then fit that razor, and then that will be operational in large testing centers like Tier 3 hospitals and also at blood centers that are run by companies like LabCorp and Quest Diagnostics.
Got it. Thank you.
Yep.
Let's go to Navneet.
Great. Thanks, Anand, for walking us through the story.
Sure.
Quick question again, along the same lines on market entry and commercialization. Sure. So, as you mentioned, the larger market and the real focus is on the clinical side.
Right.
Now, liquid biopsy and now you're adding the exosome layer on top of that. So they're not applicable to all tumor types. Like certainly the more heterogeneous tumors are less suitable for monitoring and detection via liquid biopsy technique. So any thoughts on where you're going to hone in the beginning and then go inch deep and then a mile wide, or how are you thinking about that?
I think that's a great question, Navneet, but the one thing that I would stress is we're trying to get one stage away from the actual tumor formation. So we're looking at the expression of aberrant microRNAs, either both in the cell-free fraction or in exosomes, well before the tumor is really established or even compartmentalized. The two cancers that we're looking at specifically would be pancreatic and glioblastomas, because they're both mostly silent tumors, and they show fairly homogeneous approaches in both organs, and both, when they show symptoms, are highly metastatic and mostly fatal.
Got it. Thank you.
Yep.
We still have a little time, so if there is a judge who wants a follow-up question, go ahead. Okay, great. Thank you very much, Anand and Stephen. We'll carry on. Our next company to present is Amplified Sciences. It is a diagnostics startup focused on accurately detecting and preempting the risks of debilitating diseases. The company leverages IP licensed from Purdue University. The technology is disease state agnostic, can detect to the single molecule level, scales easily to the point of care, and provides a very favorable cost structure. The lead assay is an ultra-sensitive molecular sensing technology revolutionizing pancreatic cancer. Diana Caldwell, President and CEO, will present next. Go ahead, Diana.
Thank you very much. Can you see my screen?
Yes, your time starts now.
Thank you. Such a pleasure to be here with you on a Friday afternoon. I'd like us to start by first imagining that we're a patient. Let's imagine we're having some stomach problems off and on. They're pretty severe, so we book an appointment with our primary care doctor. She thinks it's our gallbladder, so she orders a CAT scan. CAT scan comes back clean for gallbladder, but it shows a cyst on your pancreas. You immediately do some Googling and you're scared. You understand there's a link between cysts growing on the pancreas and potentially pancreatic cancer, and you soon learn that you're going to have to see a specialist and that the surgery to remove these is not routine, and it's called a Whipple procedure. So the problem that Amplified Sciences is first addressing is a huge unmet medical need and what to do about these cysts when they get picked up incidentally. That means patients aren't necessarily symptomatic because of pancreatic cancer. It's early. And if it's early, you need to do something about it. Fourth deadliest cancer in the US is pancreatic cancer. 74 out of 100 patients are dead within the first year. And the surgery to go in and remove these cysts, well, you just can't do it. It's called the Whipple, and there's a high morbidity rate, costing our US healthcare system alone $4 billion to manage these cysts. And unfortunately for patients, but fortunately for Amplified Sciences, there's a high false positive and negative rate with the competition, really in a very competitive landscape. I want to first remind you on the left that the total in vitro diagnostic market we play in, so we're a multiple product company, is $88 billion, affecting 70% of healthcare decisions. Diagnostics are. When you look at our first lead assay, PanCyst Pro, pancreatic cancer diagnostic market is over $2 billion and growing rapidly thanks to our friends in imaging. And with the 25% market share, our first product has the potential to reach $80 million. So what is this product? We use a tiny amount of the cyst fluid that is obtained through an EUS FNA procedure with gastroenterologists. It's a rule-out test. It really will be first in class, really blowing away the data from CEA. I'll show you in a moment. So we beat the competition in accuracy and overcome sample size limitations. And also great news is that we've got a $4,000 existing reimbursement pathway from a competitor that we can follow with Medicare. Here's a bit on the competitive analysis. Of course, we're the obligatory up and to the right, but we beat on accuracy as well as sample size and early detection. Here's our data against head-to-head with CEA. We're in the red line. CEA is in the blue. Actually, when we compared this in 120 bank samples that were actually validated against resected cyst, here's the high rates we got, 98% sensitivity and 100% on the specificity side. Phase 1 is a rule-out test, and then we'll scale to additional genetic markers and rinse and repeat to other disease states. We're an exact sciences model mix of molecular diagnostics in a lab-developed test, LDTs in a CLIA lab, and 510K products. I think Cologuard's already been mentioned before. They certainly are. Have gotten a lot of attention and doing a lot of advertising. Our commercial strategy is three-pronged: direct sales, focused on good news here. For a startup, we can do it. We can launch to about 70 to 80 large gastro practices in the country. Key opinion leaders and publishing over the next 2 years are critical, and obtaining that reimbursement. Our progress and path to market is right in the middle. We've closed our seed round above the minimum. We're at 1.1 million in the bank, but we do have the room to add another 100,000, which I'm still looking to hope to do over the summer. And with those funds, we'll leverage our composition of matter patents, the early data, and we'll deliver a technical and clinically validated assay ready to start processing human samples in 2022. The team is broad in its expertise. We've got a couple in-house scientists working in the lab today under the direction of my co-founder, Dr. Joe Davidson, the inventor, out of Purdue University. We've got clinical research expertise, as well as scientific, financial, regulatory, and commercial. We will succeed because we've got the right team in place, a quick path to market, and a validated market, and the right partners. We've got external validation, including this event, thank you, as well as NIH and NSF. And there's a lot of paths to exit in the molecular diagnostics and specifically in vitro diagnostics.
Sorry, Dinah, the time's up.
So join us in building a great diagnostic company focused on pancreatic cancer first.
Great, thank you so much. We will take questions. We'll first go to Sean, then to Navneet, then Derek.
Great, thanks, Anna. What jumped out at me is the $4,000 reimbursement, and it almost sounds, too good to be true, and being able to access that. So I was curious. It's not my area of expertise, but what underpins that a $4,000 reimbursement? Why is it so much? And if you're coming in and undercutting the competitor with a more accurate test, are you going to be able to justify the $4,000 sustainably for the next few years?
Sean, that's a great question. So we have a former Exact Sciences reimbursement consultant on board. We're surprised too. So that's Interpace Diagnostics, a product called Pancreatic. And so they secure with Medicare $4,000 reimbursement. They have not been successful with the commercial insurers. So we're working out our strategy right now, both publication strategy to ensure we have the data, and then also considering we've got price modeling down to what if we get an $800 reimbursement? We're still viable. Yeah. Up to maybe more in that $2,000 to $2,500 range. So we get better coverage. So we're looking at all those and trying to be the best IVD diagnostic reimbursement specialist around, because a CLIA lab LDT is a different animal than many other medical devices.
Yeah. And what I think, drawing some parallels from other, these types of diagnostics is, the Medicare folks and CMMS will try to reduce it based on your cost base, right? And so it'll be good to be aware of that as you're trying to address the TAM and whether it's worth it or not.
That's right.
One route or the other.
Right, and so to be transparent too, they are NCR and they do both NGS and PCR. Excuse me, and so we don't need PCR to do our technology. It's an optical read. So the balance of being the cost of our processing and novelty compared to them will be a bit of the strategy and dance as well.
Great, thanks.
Great, we'll go to Navneet.
Great, Diana's question along similar lines. So I know you showed clinical data in terms of sensitivity and specificity as being one of the key differentiators. Other than that, if you look at the workflow, the timing, total time to do the test, involvement of the GI, are the other tests also FNA based? Just trying to get a sense of, yes, the data is there, but you probably need more of it, but how does it compare on the other aspects?
It's a great question. Yeah. So I hate changing the patient flow or physician standard of care, right? It's easier when you can fit right in. I've been doing it my whole career. And it's really hard when you have to change. So to answer your question, the EUS FNA, there's 1 million patients in the US right now eligible for that. About a third get the EUS FNA procedure. We fit right into that. And in fact, our requirement for fluid is much lower, going down to 10 microliters. So we don't disrupt that. We could use almost medical waste kinds of fluid volumes in that process. So we don't need to change that. We will, though, because we can use smaller fluid size, we will encourage gastros. We believe over time, our thought leaders tell us, our docs do, that they can actually sample smaller cysts, so we can help grow the market a bit. We're not banking on that in our models. And so they aliquot the samples after they draw. They're in the gastro lab typically, and then send it off to the CLIA lab for processing. Typically, CEA is done in-house, but not always, particularly because the literature well documents that 20 to 40% of the time, a standard CLIA lab can't process CEA, and we'll be able to overcome that problem as well because there's too small of a fluid volume. So we overcome fluid volume issues, and then we really knock it out of the park against Interpace Diagnostics. Their current processing time for their NGS-based product is 3 weeks. And doctors tell us that's just too long when you think you might have pancreatic cancer. So our technical requirements are 3 days, but we believe the test processing day, based on the data we've already done, is less than a day. So we can process the test in a day, but we want turnaround to doctors in under 3 days or under.
Great. Thank you.
Great.
So Derek.
Yeah. So my question has to do with sort of two things that you touched on. One, it is a competitive space, and data reigns, and so that's going to prove out. But you also mentioned that this has potential to be a platform diagnostics company or multi-diagnostics company. So sounds like there's some kind of magic IP underlying all of this that allows you to be able to all sort of slots in this tool belt. So can you talk about that a bit?
That's right. Derek, I'm so glad you asked that because it's impossible to cover the platform in a 4-minute pitch. So great question. So our IP is first and second gen. It's a suite of composition of matter IP, which is unusual in diagnostics. You oftentimes are seeing process and methods patents. So we're drug-like in that route. So it's a class of dyes and a suite of dyes that Joe Davidson out of Purdue has figured out how to tag to anything. So we've tagged it to antibodies, proteins, peptides, DNA hybridization probes. It's a very flexible platform. And then we read it optically. It's secret sauce on the chemistry. And then we are agnostic, though, to therapeutic area and the reader, but our best amplification to date is Raman spectroscopy SIRS, which is a niche instrument application that's used in life sciences, but broadly. We also, though, fluorescence, plate readers, we know that we work with others, optical reads, so we're flexible on the instrument. We're not tied to a specific instrument. It's a chemistry reagent platform play.
Interesting. Thanks.
Great. Thank you so much for that, Diana.
Thanks, everyone. Have a great weekend.
So our last company that we'll be presenting is Genome Tech. It develops smartphone-size, fully automated genetic analyzers based on patent-pending optical heating technology. It can be utilized in 15-minute infection or mutation detection in personalized therapy. The company combines confidence in molecular diagnostics with simplicity and cost reduction. Miron Tokarski, President and CEO, will be our last presenter. Go ahead, Miron.
Thank you very much for introduction, and thank you for joining me for this presentation. Can you see my screen?
Yes, go ahead.
Great. So generally, all of us knows that during the first interaction between the patient and the doctor, both parties are willing to have a proper therapy introduced as soon as possible. And this is really important, mainly in infection management. Unfortunately, the accurate technologies like genetic testing requires a lot of time, and for medical testing in laboratories currently usually takes approximately 24 hours. If we will look at the market, which is growing steadily, and with the COVID-19 is growing even faster than previously, all of those technologies still require laboratory. And the INA technologies that we are utilizing in our system are growing in the fastest rate. And as I said, currently, in order to perform genetic testing, you need to have a well-equipped laboratory and skilled staff. And we are seeing this all over the world right now on the COVID-19, that the throughput of laboratories is simply too low. And generally, the hands-on time in laboratory for genetic testing is approximately 4 hours. But what is really painful is the time that passes from collecting a sample in a doctor's office or in emergency department to the time when the result is available for the doctor to introduce the therapy, as usually this is between 2 to 14 days, depending on the target that needs to be tested. With our solution, all the healthcare professional needs to do is to apply a sample on microfluidic reaction card, insert the card into the reader, press analyze, and wait for the results. We are reducing the analytical phase from 4 hours to approximately 15 minutes, and because all the steps are happening right in the doctor's office, the time from sample to result is also reduced to the same 15 minutes time frame. Our technology is patented in US. We are still waiting for the patent approval in European Union when we are based, as well as Japan, China, Brazil, and Canada. And it is based on our optical heating and detection technology that delivers the true reaction card that is fully separated from the control device. The passive reaction card is controlled only through the analyzer and has no electric connectors between the analyzer and the reaction card. We are developing primers and reagent compositions on our own, and we have also developed technology for freeze drying our reagents so that the reaction cards can be stable for up to 6 months in room temperature. And we have proven in clinical performance studies that our technology works. Internally, we have shown that for many of the pathogens tested, we can detect as low as 5 copies in a record time of just 13 minutes from the start of the amplification, and the specificity and sensitivity compared with qPCR. We can test various types of samples for bacteria, viruses, fungi, parasites for up to 5 pathogens in a single reaction card. And we have also developed commercially available in Europe SARS-CoV-2 reaction for assay for laboratories that can deliver amplification in less than 20 minutes. And when looking at our competitors, we are the only solution that delivers the all-important aspects for point-of-care genetic testing. And the vast majority of our profits will come from selling the reaction cards that we want to sell in B2B in 2022. And I couldn't be more happy to work with our team that has an experience in PerkinElmer, Novartis, and also in biomedical scientists. Now we are looking for approximately 2.5 million euros of additional financing that will lead us to clinical performance studies in EU, as well as trial production and industrialization of the analyzer. Thank you very much. I'm looking forward for your questions.
Perfect timing. We'll go to Derek first. Hang on, it seems to have some trouble in finding Derek. While we find Derek, I apologize to Sherwood from Adam Bioworks. I completely skipped over your presentation, so I'll come back to you. Bear with me. Walter, are we having trouble finding Derek? If so, we can go to the next judge, Navneet.
I think Derek's back.
Sorry about that. My internet's cutting out. I guess that means it's my turn to ask a question. So, you know, I don't know if this was asked, but if this really works as advertised, I'd be running scared if I was LabCorp and Quest Diagnostics. So, you know, that said, is this really a full replacement for all that equipment, or what are some of its limitations?
Yeah, so generally the limitations are with the multiplexing. So we are just targeting for the 5 pathogen generally. It all depends on the situation. So we are applying 5 reaction chambers for testing. One is for positive reaction. So this is a limit that we think is reasonable in order not to exceed the information that is provided to the doctor, as simply it might be they might be overwhelmed by the number of information that is available. So you always choose the panel that is intended for example, respiratory tract infections or viral respiratory tract infections or sexually transmitted diseases. This technology is in the final stage of development. It will be finalized by the end of Q3 this year, and then we will go to the stage of industrialization of the analyzer. The reaction card that is co-developed by our partners in Germany will be also ready and can be mass produced just from September, October timeframe. And for the SARS-CoV-2, from the molecular biology point of view, this is quite important that we can enlarge the throughput of laboratories without changing the equipment by a factor of 2 or 3, so at the same price. And generally in the COVID pandemic, this is really important that we are also offering something that has been tested externally and can be utilized in laboratories to help to fight with the spread of the virus.
That looks interesting. Thanks.
Let's go to Sean.
Hey, can you guys hear me?
Yes.
So my question is more around the ultimate application. So when we develop platform tech like this and razor blade model, you scale it up, and it's sounds like a great idea, but ultimately we discovered that the target area isn't as big as we thought, or what have you. So what are the maybe top 2 or 3 ultimate diagnostic areas of need that you would be pointing this great platform towards, and then associated TAMs to those?
Yeah, so really good question. And as I said previously, we are targeting for the market entry, we are targeting with respiratory tract infections panel. There will be 2 separate panels, one for bacterial infection and one for atypical/viral infections. And another panel that is also based on the market reports, quite important, is sexually transmitted diseases. So for example, for more professional healthcare like gynecologists, for example, this is very, very useful for pregnant women, for example, to have the test right in the doctor's office.
Got it. And does it directly solve an issue in each one of these, or is it a combination of other dependencies and data that you require to get to a conclusion or a diagnosis?
No, so it works just like a genetic testing. So all you need to do is to apply a sample, and there is a fluorescent detection of the amplified product, just like in a PCR. And this is, let's say, isothermal amplification. So we do not need thermal shifting. This allows us to shrink down the size of the device and lower the energy consumption. And simply that's all. The system compares the delta between the fluorescent signal at the beginning and at the end, for example, after exceeding some threshold, and issues result, for example, for specific pathogen, a specific reaction chamber.
Got it. And it's saliva that's the input medium, or is it blood.
For the respiratory tract infections, it's always better to use a swab. For example, from a throat, the material is concentrated. For the saliva, it needs to be probably pre-concentrated. So we are testing saliva for laboratory assay, but for the point of care system, it will be rather swab because it has higher concentration of the material. And the same applies for sexually transmitted disease.
Got it. Thank you.
Go ahead, Navneet.
Miran, so very cool technology. If you can cut something down from a week or 2 to 14 days to 15 minutes, that's awesome. And we always see these with point of care diagnostics. They're all very exciting and they cut down the time significantly. But where when we start to dig deeper and where some of these run into challenges, and want to get your thoughts on it, is from a cost perspective. Right, so it's great that you cut down a 3-day test to a 15-minute test, but now are you converting a test that costs $50 and would give a result in 3 days to a test that now costs $500, giving a result in 15 days, whereas those are 15 minutes, and whereas the payer and the provider, from the payer's perspective, they're okay with the 3-day wait because you get a conclusive result, so they don't want to pay $500, and from the physician's perspective, they don't want a box sitting in the room. They're actually okay, writing a script and sending it off to a third-party provider. So have you thought about that? Like, how does that work especially from a cost perspective? You convert something.
So generally, for this is a really great question. So from the perspective of point of care, in order to the solution to be widespread, you need to have price competitive entry point for the system. If the system costs, for example, $20,000, it won't be point of care because you need multiple devices. So this is why our technology enables to shrink down the size and also cut down on the costs of such devices. The production will, the final cost of the device will be approximately $2,500. So this is, let's say, 10% of what is offering the Abbott, for example, or Roche with Cobas Liat for flu testing. And for the reaction card, is also important that we have a passive reaction card without electric sensors, heaters, etc. So the price of the reaction card is also lower. And we are not going into syndromic testing, like, for example, QIAGEN is offering some solutions that they are testing for 20+ pathogens on a single reaction card. We are just focusing on 5 in order to shrink down the size of the reaction card and also cut down on the costs, so that it will be below the current CPT codes for genetic testing in US. And also for the doctors, if this will be possible, of course, it depends on the market, it can create a new revenue stream for them. So generally, now they refer patients for genetic testing in the laboratory, and they pay, for example, $70 for this. Whereas there is a profit generated in the laboratory. We can bring this to a doctor's office so that they will generate the profit, and the final price will be within the limits of CPT code.
Got it, thanks.
Thank you very much, Miran. I appreciate it. I wanted to apologize to Sherwood Yao, who was supposed to go before Miran. I'm going to chalk this up to this being Friday. So we will now hear the presentation from Atom Bioworks. This is a company that's creating a new platform for precision antiviral medicine. Its pattern matching enabled sensing and therapeutics is a disruptive technology that uses synthetic DNA to construct DCE-specific pattern matching nanostructures to identify viruses and provide treatments. The company's current product portfolio includes a diagnostic device that can match the performance of the gold standard PCR tests. Sherwood Yao will present next. Sherwood, your time starts now.
Thanks. Thanks, everyone. I guess we're the best secret company on the panel. So our product is Atom Bioworks that developed this instant diagnostic. We're pushing the 30 minutes even further. So how can we get it done? But there's a pressing need for that. Today's diagnostics, in particular for the COVID, we have seen a lot of those testing. They're doing the same thing by different companies. They're putting those cancer targeting devices like ABI 7500, and they're put a 45 minutes test into those systems. but those systems will not work for point of entry. So what is point of entry? So look at airport, cruise line, schools, where every business now needs to open. They have to have a faster way to test it. And this 45 minutes is not going to cut it. So what we're working on is actually a 1-minute test. And this is our super fast and sensitive test. We were founded by NSF, SBR. So on the left, you see our results. We can get close to 1 minute. And this is faster than Abbott's ID now. We're able to beat their performance. On the right-hand side, you see our high sensitivity. And there we can detect OC43. This is where it's a sibling of coronavirus COVID-19. So we're able to differentiating and be able to detect 100 copies per mil. And so this is where Matt from ResearchDX, they have done 9 FDA EUAs this year for their clients. And he said this will disrupt the market. So how are we able to do that is we have this polyvalent binding nanostructure defined by software approach. This research was featured, our research was featured on Nature Chemistry, Times, Scientific American. So what that does is we're using AI and software to design this DNA nanostructure. This star stain is per pattern built to match the disease target. So this we're looking at the antigen of the virus, of the protein, and once we find a pattern match, we're able to bind it so strong, we're able to produce very strong signal. Our approach works with a wide range of body fluid samples that include saliva and nasal swab. And once the patient sample with our assays mixed, it took about 1 minute to mix and to generate the fluorescent signal. So the signal is generated by the fact that our sensor is pattern matching to the disease target, and then it's only matched to the specific target. It won't match to anything else. And if it finds it, it will emit the fluorescence. This fluorescence then allow us to develop this assay that can fit into a lot of existing systems. Plus, we're able to get into a whole range of connected systems, such as smartphone. And then, by the way, we can also do instrument-free tests. And also for our point of entry testing, we need a high throughput test where we want to test hundreds per batch. And so to have such an assay developed, and it's very fast too. So our software-defined approach allows us to design candidates and going through a screening process to produce such an assay very rapidly. So such a clinical diagnostics, we have started with a few infectious disease targets, and we are going through a few more. And here, our COVID-19 target has going through our CDO partners. We're going finishing our clinical validation and going to FDA. And we have already raised a few rounds of early investment. We're seeking $5 million to generate $10 million in next year. We're going to bring this product to the commercial labs first. And then during this time, our R&D will focus on the point of entry development. Our team has a lot of IVD veterans. They came out of LabCorp, New Genomics, and all those companies. And in summary, our product is sensitive, it's fast, it's less than 2 minutes, and it's very easy to use, and we don't disrupt today's workflow. And it's rapid to deploy. And that's us, Atom Bioworks.
Great, thank you very much. Let's go to Navneet for questions, please.
Great, thanks Sherwood. Very timely and relevant topic here and product, of course. So quick question for you, and this might be, I may not have understood the value proposition entirely, so pardon me for my naivety there. So the 1 minute versus 45 minutes, makes a lot of sense at point of entry. But when you come down to, let's say, 10 minutes for the Abbott test versus 1 minute, now if I'm thinking about my airport point of entry experience, if it's all about just figuring out operationally the workflow, would you bet against Abbott not being able to figure, if that's the test that's available right now and you guys have a year or two to come to market, for them to figure out, all right, because the sample prep time, if I recall, is the same. It's about how long it takes to get the reaction.
No.
Right. So if everybody takes a test at a point, and from my experience, yours might be different, it takes 10 to 15 minutes at least to walk around the airport, get to customs, immigration. By the time you have the test, the results, how does that, are you getting what I'm saying? Like, does that nullify some of the benefits going from 10 to 1, or is there some other application that I'm not understanding clearly?
Yeah, so fundamentally, that Abbott's ID Now product is a single threaded test. So that instrument can only allowed to have one tube to fit in. And so that device is really a point of care device. It was meant for doctors to allow them to treat or screening the patient. And that's a small volume compared to the airports. So in the airports, we will be working with some robotic companies and to have this very high throughput system. So those systems will allow us to check about 200 people, because 200 people in 2 minutes. So that kind of a volume is much different than what a single threaded 13 minutes can do.
Thank you.
Let's go to Derek next.
Yeah, so, interesting proposition here. And it's more than just airports. I would imagine, any business right now, or many of them are checking temperatures in your forehead. And so if you had the equivalent kind of thing to do with the COVID test, obviously everyone would want that. That being said, do you think it would be adopted? Because there's so many, the results aren't always accurate if for these tests, and if something is gaining on time, is it trading off somewhere else? So it's kind of a two-pronged question. Is this better than the Abbott test, not just in speed, but also in accuracy? And do you think that, I guess that kind of answers both questions.
Sure. And so that actually has two parts of our business. So our part one is we're working with today's largest lab. So one of our founding team, he used to work at LabCorp, and now that team, he has now building a team to building the material and build the whole companion diagnostics for those large labs. And so the initial business, our first revenue generating business, is going to become a material supplier business. So we're building assays that's going to fit into today's lab existing workflow. We're not disrupting anybody's business, and it's only making their profits goes up, because today, for example, companion diagnostic takes about a day to finish, at least a day. And so if we are able to turn this around within a few minutes, and then this increases their profitability. And so this is where our CDO partner is very excited to bring this to their existing business. And the second is, where does this bring to the existing kind of, if I can use a thermometer and check the forehead, will that be good enough? Obviously, if one of the charts on my second or third slide, it shows the sensitivity and the specificity. And that shows actually we are able to get to about 100 copies. So what 100 copy translates is the first day or second day of the infection. And that means that that patient is able to spreading the disease. And so if we are able to detecting that, and there's no way anything other simpler stuff, for example, the temperature check, they will not reveal that kind of patients. And so we definitely need more rigorous testing. That's where our current clinical testing is going through that process. We're trying to decide and find out where are the loopholes that our detection is going to miss. But we're also positioned us as a screening test. It's not a diagnostic test for the final treatment. I hope that makes sense.
Mm-hmm. Yeah, thanks.
Okay, let's go to Sean.
Hey, Sherwood. My question is around your AI part. So it sounds like you can also recognize virus patterns that are new. So are you able to then leverage the technology to look at variations and mutations in the virus, in addition to the current known ones that you're detecting at a high volume?
Yes, so this is one of my chart shows that we're using the AI process to fine-tuning our sensors to be able to detect, for example, different subtypes of coronavirus. And so we have two levels of control. One is on the geometry, the spatial arrangement, that star, and the other is at the binding spot of the star. It has all the ligands. So those two factors allow us to have a very fine control on what this thing can target to. And by tweaking those two parameters, we're able to making the sensor select just one, not the siblings.
Got it. Okay. I think I get it. It sounds complicated, but I think I get it. So thanks very much.